a gente recebe o professor Solaiman Shokur, da Escola Polytechnique Fédérale de Lausanne. O professor Shokur é um neuroengenheiro, né, durante toda a sua carreira, e tem um contato bem próximo com o nosso instituto, trabalhou no projeto Alcagen, fez uma visita também durante um período da sua vida no laboratório do professor Nicolelis, na Universidade de Duke, e atualmente é professor na EPFL, de novo, né, na Escola Politécnica Federal, também trabalhando os mesmos temas voltados para neuroprótese, né? E aí o que ele vai falar hoje para a gente aqui é basicamente sobre um approach modular ligado à neuroprótese, ao desenvolvimento de neuroprótese. Bem, espero que vocês todos aproveitem essa palestra do professor Solaiman, certo? E aí no final a gente começa uma sessão de discussão com perguntas, certo? Então, aproveitem bem aí e boa palestra.
Muito obrigado pela apresentação, Ramon. Muito obrigado pela organização. Ramon, você pode deixar sua câmera aberta, só para eu ter um feedback, no caso, vocês estão me ouvindo? Alô? Porque nesse momento eu estou sem feedback. Alô? Você pode ficar, obrigado, com a câmera. Então, bom, my presentation is going to be in English. This was the limit of my Portuguese at the moment. Gonna share my screen in here. Just please let me know if you can see my screen well. Can you see my screen and hear me well? Okay, thank you very much. Thank you. Well, again, thank you everyone for the organization, and thank you for inviting me for this symposium. My name is Solaiman Shokur. I'm working at the Translational Neuroengineering Laboratory here at EPFL, and this presentation, the theme of this presentation will be this modular approach for neuroprosthetics. Practically, in the coming 20 minutes, I will be giving you some practical examples of neuroprosthetic solutions, which are meant for very different different patients, and pathologies. For example, bionic hands used to restore sensory motor functions in amputee patients, or for example, bidirectional brain-machine interfaces, which is more classically used for stroke or spinal cord injury patients. And what we will see is that a lot of these technologies, they share, although they are generally studied by very different groups and very different even laboratories sometimes, they actually share a lot in common. And it's possible to sort of benefit from the knowledge from one to use it for the other one. So hopefully this will be interesting for the new or future neuroengineers that are in the room, and maybe also a new take on for the more expert or professors that are in the room, looking at this real crosstalk between apparently very different neuroprosthetics, but seeing how much similarities there are among them. And for that, I'll be using concrete examples from a bionic arm development that we do in the laboratory where I'm right now at the T&E lab, as well as examples of bidirectional BMIs, or brain-machine interfaces, that I developed when I was with Professor Nicolelis, as I'm nicely presented at the moment. And for that I will be using, so my understanding that this is a neuroengineering symposium, so I'm guessing there's a lot of engineers in the room. So for that I will be using an analogy for, I think, what was probably our first setup as engineers in our life, which is Legos. So we will be using the idea of Lego blocks and building blocks, and see how we could imagine future neuroprosthetics, future generation neuroprosthetics, using the knowledge that we already have. So, and a lot of what I'm going to tell you today, by the way, comes in this review paper that that was published a few months ago in Med on this modular strategy. So, a neuroprosthetic could be defined with several fundamental building blocks, which are blocks for motor decoding, blocks for movement restoration, and blocks for sensory feedback. I'll be talking here about neuroprosthetics to restore sensory motor functions. I will keep apart prosthetics for cognitive neuroprosthetics, but let's say all the prosthetics that are meant to be restoring sensory motor functions use this sort of fundamental blocks. And these fundamental blocks are made of a number of what we call neurotechnological modules, which we have classified here in 6 subclasses, which all of the solutions have some implementations of the subclasses, which are recording modules here with, for example, electrodes that could be invasive or non-invasive. For example, EMG, non-invasive EMGs for peripheral nervous system, or EEG system for the central nervous system, or ECOGs, or implants in the brain, or let's say Utah arrays, name it, any sort of electrodes that is interfacing with the nervous system to record signals. Second, we have a module for extracting motor parameters using some sort of algorithmics, some sort of ways to extract features and decode them, running on some hardware which could be embedded, or maybe a robotic hardware, or excuse me, a simple computer. We have next modules for movement restoration or actuation, which could be robotics, or again, electrodes that could interface with the nerves as functional electrical stimulation, for example, or even electrodes that interface with the nerves. We will see a few examples of that. And finally, blocks to sense, encode, and stimulate to then restore sensory feedback. So here we will have classes of, for example, sensors that are coming on top of a robotic hand. We have algorithms here for encoding sensory feedback, and then the hardware is generally a stimulator, a pulse stimulator, using again an electrodes that could be invasive or non-invasive, that then will interface with the nervous system. And you as a neuroengineer, you will sort of specialize generally into improving one of these blocks. So the goal will be either to look at these blocks as they exist and come up with a new implementation. Rarely you will be involved in making all the parts of a neuroprosthetics. And more classically, a laboratory or group will be specialized in improving one of them, maybe come up with an amazing new algorithm for decoding, or maybe coming with a new way of coating the electrodes to improve the impedance to make it the signal better. And what we also start seeing very quickly already here is that improvement in one of the domains could benefit many others. So if you have an improvement on the electrodes, so if you do a better signal for electrodes with a higher, let's say, biocompatibility, it could both improve the motor decoding part, but also the sensory part, and in some cases also for the motor restoration. So now we will jump into some practical examples with that. And I will start here with the bidirectional robotic hands or bionic hands. Here is an example of what we do routinely here in the Translational Engineering Lab here at EPFL, mainly with the impulse also of Professor Silvestro Micera, who is the head of the laboratory. So in this example, published in Science Translational Medicine by Rojas Popovic and colleagues, there is amputee patients with surface EMGs recording muscle activity here that are then decoded to control the motor commands of this robotic hand. This robotic hand has embedded sensors, which are pressure sensors in this case, which then here are translated into current, into stimulation current, using a relatively simple linear function which looks at the pressure function and translates that into stimulation amplitude using a simple a saturation function. Obviously, you don't want to go crazy high in your stimulation if the forces are very big. So this readout of the sensors is then translated into this stimulation parameters, which then is injected into electrodes that are implanted inside the nerve. These are called time electrodes. I will show you a bit more in detail in the next slide, which are implanted in the medial nerve and the ulnar nerve of this subject. This subject is a transradial amputee patient, and the subject now can, with the eyes closed, control a robotic hand, touch different objects, feel them, and then experience, also have a closed loop control on that to recognize different objects or shapes. Let's look now into some of these modules here. On the motor decoding part, there is a million kind of different algorithms. I'm sure people at ESD are already familiar with that. So the most classic solution for decoding is a simple thresholding decoding. This is going to be probably your first approach as a student, recording EMG signals, looking when the signals, for example, I'm recording one muscle, and I look when the signal goes above a certain threshold. I'm sorry, the green light, I think here is wrong, should be slightly below. And whenever it goes above this threshold, we say activation done, and that will control a robotic hand to, for example, grasp. Now, more advanced solutions uses machine learning. In the case of Raspopovich and colleagues, or what we did in the lab, using multi-layer perceptron here, so an artificial neural network. So we don't use only one electrode of EMG, we record a bunch of them, in this case, 4 of them. We extract the interesting information from the signal, and then we use a classic classification solution, as I said, artificial neural network, but there is SVM, deep learning, there is really kilometers of literature about that. And on that review, if you want to look at it, we are listing a lot of this algorithms that are being used. In this case was a 5-class classification, so detecting when the subject wanted to rest the hand, a palmar grasp, and different kind of grasp, like the pinching or like cylindrical grasps. As for the we also wrote a review about this a few months ago with 2 of our students in the annual reviews of controls and robotics, where we really tried to list every single robotic hand that we could find in the last 25 years. We selected 75 of them and then give the details among those that are commercially available, but also those that are more research-based prosthetic hands. Looking at one very important parameter here, which is the degrees of freedom. So how many joints could move on the hands differently, how many motors you have. And generally speaking, in these robotic solutions, you have what you call an under-actuation, meaning that you have less motors than degrees of freedom. Let me say that simply, if you're moving this finger here, you probably don't need 3 motors for each one of these phalange. You maybe only need 1 motor, and the rest of it will be done to some clever mechanics, making sure that then the finger will come around an object, but then you have only 1 degree of actuation for 3 degrees of freedom. And that's what describes here the degree of actuation. Again, if you're interested, look at this list. You have all the examples and then the description, if they have or not sensors embedded in the hand, which kind of motors they have, etc., etc. On the well, in this case, we use the Azura hand by Prinsila, which has nice sensors embedded also in it. So this was the choice for the Raspopovich paper. For the electrodes, we used, so there are also a number of different electrodes in the literature. I'm listing here 3 classic one used for interfacing with the peripheral nervous system. Cuff electrodes, which are probably the most stable one, the one that has been developed in the 70s. So we have a lot of knowledge about it. So these electrodes go around the nerves and then has 3 to 4 active sites to stimulate or to read from that nerve. Live electrodes goes through the nerve and then gives you a bit more of this active sites or recording sites. And here the transverse intravascular multi-channel or time electrodes, which goes through the nerves and then permits to have contact with multiple fascicles inside the nerve. Here in this case, I believe it's an ulnar nerve. Here you have on each of these electrodes, you have 8 active sites where you could read or write. In the case, in our lab, we use routinely the time electrodes, which is for us the one that gives us the most freedom because it gives us the highest selectivity. So we really be very precise. Here you're outside the nerve, so you have to put a lot of current and you're not really certain of where you're stimulating. With the time electrodes, we have a much better selectivity. But there are also many other examples, again, that you can look into this reviews and find many other examples in literature. Now, for the sensory encoding, what was used here was a relatively simple encoding, looking linear encoding, as I said, between the force sensors and the stimulation, the stimulation amplitude. The test started with a little characterization where the experimenter stimulated different active sites, and then the subject has to say where he or she perceives the stimulation. So you stimulate, for example, active site 15, and the subject says, I have the feeling you're touching me here, or maybe I have this sensation of a tingling in this finger. So you first build your map on that. Next, what was done here was, so the subject was controlling this robotic hand and touching objects that were hard, medium, or soft. Here is the readout, I guess, again, of the sensors, and here is the current amplitude of the stimulator. And what the experiment showed that was the subject were significant, were able to detect this, the object stiffness significantly above chance, which in this case would be 33%, obviously. So they were able to detect it based only on the stimulation. On the other hand, they also, the tests were done with shape recognition. So here testing different object shapes, cylindrical, small, or spherical. Again, subjects were able to detect them with significant accuracy. How is that even possible is because the time electrode permits you to encode sensation on different hand parts. So this is not just an on-off. It's not just that you have one only encoding in him. You only know when it's touching or not touching. Not only you have that, but you also know which part of the hand is touching. So if you're grasping, and if you do the test by yourself, you're grasping an object that is cylindrical or spherical, you will see that you have different numbers of fingers that are in contact. So this permitted also the subjects to have a relatively intuitive understanding of which object they were using and what was its stiffness level. Now, jumping into the second part of this presentation on bidirectional brain-machine interfaces, here I'm going to take some examples of what I collected, some data that I collected with Professor Nicolelis on rhesus monkeys. Here is an example of a rhesus monkeys with brain recording. The electrodes I will show you in a second. I'm sure you're going to recognize it. The monkey trained with a brain-machine interface to control an avatar arm that appeared on a screen that touched different virtual objects and then received the sensory feedback through a stimulation in the somatosensory cortex, so directly in the brain. Here, for the motor decoding site, we used this sort of electrodes. I'm sure that people know how to build even that at the ISD in Macaiba. This was developed by Professor Nicolelis and colleagues in the late 90s. So these electrodes are implanted in the M1 and S1 bilaterally in this monkey. And the decoding of that was the very classic 2003 Carmena protocol, Carmena and Nicolelis. So a linear decoding, the end effector position and speed was a linear function of the frequencies of the neurons that were recorded in M1. In this case, 96 neurons recorded in M1 thanks to these electrodes, and which permitted to decode the position and the speed of the end effector. On the encoding side, or the sensory encoding side, here we used intracortical microstimulation, so small charge balanced stimulations directly in S1 in area 3B for neuroscientists. This part might be obvious. Why? Is because that is the part that encodes for vibration and is time locked to this sort of vibrotactile sensation. Now, I have an example here of what the monkey is seeing and how the experiment goes. So the monkey controls this hand, and you will see that two objects appears, and this object have different textures. And these textures are represented by their let's say, spatial frequency of the ridges of this texture. So you have objects with higher textures and objects with lower frequency textures. And the monkey has to move the hand on the two of them. So brain control the hand to move on that. And then every time the fingertip of this avatar touches one of the ridges, he will receive one pulse of stimulation, right? So the monkey sees this. He does not see this. And I'm going to just let it play a second. Okay, as you can see, the monkey is sort of exploring this object. When it's like this, he went several times on the two of them. And up to these 3 tests that we saw, he was very good at recognizing the right one, then goes on top of it and selects it and gets his juice. So let's say on this task, the monkeys learned to do this task, and they were quite good at doing it. We tested many different combinations of a couple of stimulations. So looking at the variation of spatial frequency. So for example, having 4 ridges per centimeters versus, let's say, 0.5 ridges per centimeter. Monkeys obviously were better when the difference was very large, as in here. So when we had like large difference, so these are 2 different monkeys, and this is their ability to find which one was the correct one. So if the difference between the 2 object texture was very large, they were good at it. If the difference was getting smaller, they were a bit more confused. And this followed nicely, for those of you that are familiar with psychometric measurements, followed nicely the Weber-Fetcher law, meaning that they were better at discriminating 0.5 and 1 compared to 3.5 and 4. Okay, so the differences there of what we call the just noticeable difference is depends also of the base stimulation. So just to say that somehow it also follows a natural law of how we use the other perception in our body. Okay, so now, as I think my time is running short, let's go to the conclusive part already of this presentation. So we did this work on many different examples of bionic hands here by George and colleagues, or here by Dana and colleagues, and different examples of bidirectional brain-machine interfaces. I just show you one example with monkeys. This is also being done today clinically. Here's one example at the University of Pittsburgh, and this is an example by Ganser and colleagues. So we start looking at this different examples, and all of them are built, even if they are, as I said, different sort of applications, they have the same basic construction with the same building blocks. And we started looking at these building blocks, and many of them are, a number of them are similar among them. So here, if we look at this block, which is the Utah arrays, which is the electrodes used for recording in this study, was the same one that was used in this other study for stimulation for sensory feedback. So the same module used here, for motor decoding is used here for sensory feedback. While in this other study, a very similar building block, a very similar technique, the Utah slanted electrode was used to be implanted inside the nerves to give sensory feedback for an amputee patient. So again, this similar technology is being used in many different applications. We looked for many of them. Again, I'm not going to go into detail. You can see of many of those examples in the publication that I told you. We looked at these technologies and how they were used for motor decoding, movement restoration, and sensory feedback. I'm going to individuate one example, which is the time electrodes, which I just showed you in the Raspapovich paper, where it was used for sensory feedback. So the time electrode was implanted in the nerve for the sensory feedback. But in another study, the same time electrodes were recorded were interfacing with the motor fascicles in the nerve, and they were used to decode hand movement. So in this case, they were not interfacing with the sensory fascicles, but with the motor one, and they were used here to decode different hand movements. And finally, in a newer study, which is unpublished yet, this is just an SFN paper. The paper is going to come in STM, Science Translational Medicine, in about 2 weeks. The same electrodes were implanted inside the motor fascicles, but used to stimulate the motor fascicles instead of reading them. And this was shown to restore hand movement, grasping movement, in a tetraplegic monkey. So now you stimulate the nerve instead of reading from it using the same technology, and you can restore hand movement. Again, many examples of that. Here we have nice examples of how the encoding algorithm, so if you're more from an algorithmic point of view or a computer scientist, how the same encoding algorithm have been used for sensory feedback with groups that were working in the central nervous system and groups that were working on the peripheral nervous system. So the amplitude modulation or frequency modulation of the stimulation, or what is called biomimetic spike train, this is a bit more complicated, but the same algorithms were used using different kind of electrodes. So the electrodes, it doesn't even matter, but they were used both to give feedback through the brain or to the nerves. So once again, once you have a breakthrough in one of these domains, it could be used by researchers in another domain. And I think this could be maybe interesting for future researchers. We have, can somebody from the organization tell me if I have how much time or more I have, so I know how much I can go into detail on the challenges? Do you have 3 more minutes? Okay, I assume I have time. Okay, so I'm gonna just detail you some of the challenges that we have identified that, which again is maybe interesting as a researcher working in a domain, looking at that and looking what might be the next challenge that you might be working on. So here we have listed those challenges that we perceive that could be the unmet technologies that we have at the moment and how they could benefit both for motor decoding, movement restoration, and sensory feedback. So what we need, we need many things, but these are those that we believe are interesting. We need compliant and stable electrodes. So electrodes that could be implanted in the brain or in the nerves for a very long time. And for that, we need fabrication of soft interfaces, for example, electrodes that are not hard but soft. That is exactly what, for example, Neuralink is trying to do, and some of the researchers of my colleagues also at EPFL are doing. Fabrication of ultra low footprint neurointerfaces, so neurointerfaces that could be implanted in the brain that are not they don't have anymore the connection with outside, but they could be implanted under the skin. We need also high number of channel electrodes. This is something that Professor Nicolelis has been promoting for a very long time in his career and has been really at the cutting edge for a very long time. Other researchers should be working on that for going for very high count electrodes that are implanted, so covering large areas of the brain or of the nervous system. We need low power, low voltage stimulation. Once again, the neuroprosthetics today are slowly getting out from laboratories. So far, we are still doing neuroprosthetics with huge Plaxon systems in a laboratory. This is slowly going outside the lab and even in patients' homes. So we need electronics that is easy to implant transcutaneously, that has low power, so also battery doesn't become a huge problem. We need a selective interaction with specific part of the neural population. As I show you, the time electrode, for example, is a very good solution for have better selectivity at the nerve level. We need stable interfacing. Maybe optogenetics could be one of the solutions in the brain. This is something that also has been studied in the last about 10 years. And finally, we need natural human-machine interaction. We need rich sensory feedback. We need better ways of encoding sensory feedback and long-term stable motor decoding. I myself, I'm humbly working since few years on this part of the spectrum. I'm a bit less of a specialist I'm definitely not a specialist in the rest of it, but I'm a bit more working on this one. But as I said, neuroengineering is a very broad domain with many different expertise. So many different brains have to come up with solutions, we believe, in this key critical problems in the coming years. Now, if you want to know more about this, I'm sorry if the presentation has been maybe a bit quick and hopefully not too messy. If you want to know more about it, you can look into this bibliography here, this paper that was featured a few months ago in the cover of Med. We also have this other review paper in the annual reviews of control and robotics, and another paper that came out actually about 15 minutes ago. That was I got the answer right before this call. So also on the neural resources for neurotechnology, but for human enhancement, and this came out in Nature Machine Intelligence, as I said today in a few minutes ago. Also, if you want to follow, if you want to know a bit more what I'm doing myself, what we are doing in the lab, I try to be a bit up to date on my Twitter account, so feel free to follow there. I try to also to give a bit of the feedback of the novelties that are happening in EPFL and some of the new neurotechnologies that I've been seeing around. As I'm also on that aspect, a bit of advertisement here. We are hosting, editing a special issue on neuroprosthetics and how to get neuroprosthetics out of the lab in frontiers of neurorobotics. So, well, I added this little QR code here if you want to scan, if you have a paper that you're working on neuroprosthetics and how to get neuroprosthetics out of the laboratory. It could be from the clinical point of view, the experimental point of view, the neuroengineering, electronic, name it, even maybe occupational therapies that is working on that. Feel free to contact us and to send your paper. And with that, I am done with my presentation, and I hope it was interesting to you, and I'm waiting for your answers.
Muito obrigado pela palestra, Solaiman. E agora a gente vai abrir um pouquinho de espaço para perguntas tanto em português quanto em inglês. Imagino que o professor Solaiman escolha a sua língua para responder, mas ele tem um contato bem forte com o Brasil, e então ele entende bem português, por mais que ele ache que não entende, ele tem um português perfeito. Já tem algumas perguntas aqui, Solaiman, eu vou ler uma para ti. E a primeira pergunta foi do Daniel Schultz. How fast is the sensory feedback loop?
In which of the examples? Well, I can give on both of them. So on the bionic hands, it is about 20 to 30 milliseconds feedback loop. On the brain-machine interface one, we had a funny thing there that we were playing. So when you do the stimulation, I didn't speak about that, but when you do a closed loop BMI, you start brain-machine interface, you have infinite complications there. When you stimulate an S1 and you read an M1, now you don't read in M1 anymore. You're just reading whatever you're stimulating, because you're probably saturating your amplifiers. So you cannot stimulate and read at the same time. So what we did, it was a little multiplexing. We were reading for 50 milliseconds and we were stimulating for 50 milliseconds. And we were alternating between the two. So I guess your resolution could be, in this case, 100 milliseconds. And that was enough for this case, at least for the monkey to perceive the sensation. And also for the subjects to perceive it. For the amputee subjects, as I said, 20 to 30 milliseconds.
Muito obrigado, Solaiman. Tem mais uma pergunta para ti, aqui do Fabrício, Brasil. Could you share some more information about the movement restoration of the monkey with tetraplegia after the nerve stimulation? Do they move the hand during the stimulation, or they could do also after the stimulation?
Excellent question, muito obrigado. Vou responder em português isso aqui, vai, que eu vou tentar. Esse estudo aqui com os macacos, né? Então, esse aqui é primeiros estudos com estimulação dos nervos para recuperar movimentos. Então, o macaco, nesse caso, era uma tetraplegia aguda, certo? Era de, como sabe, com lidocaína para parar 3, 4 horas. Então, a gente não consegue saber qual que é o efeito a longo termo disso aqui. Sua pergunta é super boa, porque tudo que eu mostrei hoje foi mais sobre a restauração do ponto de vista de, como falar, de bypass, ou de assistive device. Porque você está perguntando quais são os efeitos de reabilitação a longo prazo. Então, a gente tem alguns estudos mostrando que a longo prazo dessa estimulação, por exemplo, com FES, Functional Electrical Stimulation, foram super bons. So, paciente paraplégicos com estimulação funcional elétrica superficial da perna, depois da estimulação de algum mês de estimulação, recuperou algum nível de funcionalidade até quando a estimulação estava off. Nesse caso aqui, eu não posso te responder ainda não. Acho que a gente vai ver a mesma coisa, mas o primeiro estudo, como eu falei, o paper vai sair daqui a 2 semanas lá no Science Translational Medicine. Obviamente, vamos continuar mais sobre isso, porque é muito melhor, na verdade, que o FES. Porque permite com FES é difícil fazer uma restauração de grasp. É muito complicado, porque tem fadiga do músculo, etc. Com esse aqui era possível, então acho que é uma técnica interessante para ver. Espero que eu respondi.
Muito obrigado, Solaiman.
Obrigado a você.
Legal. Solaiman, a gente não tem mais tempo para perguntas, porque a próxima palestra já vai começar, tá? Mas eu quero te agradecer aqui pela participação no nosso simpósio, tá bem? E, enfim, foi muito interessante ouvir o que tem trabalhado aí na EPFL e muito obrigado novamente pela participação.
Obrigado a vocês.